Thank you very much for that introduction, and thank you to the Bell House for hosting me tonight, and also to all of you for being here to hear my talk. So, the mysterious inner workings of the mind and brain have captured people's imagination since the beginning of time. And sometimes those mysteries are made clear through observation or introspection. For example, this is Empedocles, the Greek, who wondered about how vision worked, and he hypothesized that there were visual rays that came out of the eyes, that is, light left the eyes and bounced off of objects in the world, and that's what made it possible for us to see. And other mysteries of the brain were made clear through disease states, such as epilepsy, where the sufferer temporarily loses both consciousness and motor control. And still other mysteries were made plain by mind-altering substances such as poppies and also alcohol, the use of which has gone back as far as 5,000 BC, at least as documented as far back as 5,000 BC. And realistically, it might have been a little further than that. But beyond the documented talks of the Greeks and the observations of medical doctors, there were doubtless many conversations around dinner tables and going even further back, dinner gatherings on the ground about what is it that makes us who we are and why we do the things we do? For example, why is my sister always optimistic even in the face of disaster while my brother is perpetually gloomy? Or why did the neighbor up and move in search of greener pastures when it would have been smarter to stay put? Or why did my father store only a small amount of food for the winter when many long cold months are expected? And until recently, one could really only get to the bottom of these issues or try to make headway on them at all by using things like introspection and observation, because that's really all that people had. But now, we are at a really exciting moment, because for the first time, we finally have the tools that make it possible to really answer those questions. We have tools that allow us to measure the responses of neurons, to perturb the activity of groups of neurons and look at the effect on behavior. And taken together, these techniques allow us to bridge that gap, to understand what it is in our brains that gives rise to the things that we do. So for people like me as a systems neuroscientist, that's our goal, to understand the neural circuits that give rise to behavior. And part of our motivation for doing this is intellectual. That is, we think it's an interesting problem, and as many people have going back many centuries, and for the reasons that I described a moment ago. But also part of our interest comes because this is a very important clinical problem as well. This image is a painting by Van Gogh showing somebody who's depressed. And it's currently estimated that 1 in 17 people suffer from a serious mental illness and that 1 in 19 people suffer from addiction. And this causes a tremendous cost to our medical system, and also as anyone who's been a sufferer of any of these disorders or who has been close to someone who is, this is a tremendous personal cost as well. Going through mental illness is a horrible thing to go through. And our hope is that by understanding the neural circuits that give rise to behavior, we might one day be able to inform clinical treatments for people who suffer from things like depression and addiction and so on. So let's for a moment just kind of take stock of where the field is today. How are we doing as neuroscientists in terms of understanding the neural circuits that give rise to behavior, to reaching this goal? Well, in terms of sensory motor reflexes, we're really doing pretty well. So this image here shows what's called the knee-jerk reflex, and you've probably all done this at the doctor. He hits you on the knee, and your leg goes out like that. And we understand that reflex completely. It's pretty simple. We know all the neurons involved. We can perturb it. We can do anything we want. It's solved. We're done. Another interesting example is a reflex that's really, really important, and all of you have it, but probably none of you know that you have it. And it's called the vestibulo-ocular reflex. And what it does is it keeps your eyes in your head stable so that images look stable in the world. So I'm going to give you an audience demonstration now so that you can convince yourselves that you have a VOR, and you'll appreciate it now for the first time. So what you need to do is, let's see, I'll put this here. Take your finger and hold it out in front of your face about 12 inches or so, and move it back and forth about like this. And just try your best to follow your finger with your eyes. Try to track it with your eyes. And how does it look, blurry or focused? You guys are... How many drinks have you people had? Jesus, it's like 8 o'clock. Anyway, it looks pretty... I would say it looks fairly blurry, but for comparison, let's be empiricists here. Let's make a comparison. Now hold your finger steady and move your head back and forth. And how does your finger look now? All right, the drunks in the audience, everything's blurry for them. But for the rest of you, It probably looked very focused. And that's because every time you move your head in one direction, your eyes move in exactly the opposite direction in just the right amount to compensate for that head movement. And because of that, the world looks stable. As you walk around and move your head and body around, your eyes compensate perfectly so that the world looks stable and it doesn't look like those movies that we take with our iPhones where everything is bouncing around. So we understand this reflex, the VOR, also very well. It's a simple circuit. We know all the neurons involved. We're even at a pretty good stage in understanding how it adapts. It's very carefully calibrated, so it changes throughout life. And when it comes to the VOR, I wouldn't say it's completely solved, but we're really making a ton of progress. And these are two examples of what I would call sensory motor reflexes. And as a field, we've really made a lot of progress. Things are going well. By contrast, when it comes to more complex behaviors, we're really at a total loss. And people are really interested in understanding complex behavior for the reasons that I described. And so anytime there's any kind of study about it, it definitely gets a lot of press. But when I show you some of the press that comes out about these studies, you can see why I'm kind of skeptical about how this part of the field is really going. So here's an example from the New York Times. Okay, so the person who conducted this study that was written about in this article, they took a bunch of human volunteers and they put them in an fMRI and showed them their iPhones in various states. Sometimes they had sound or didn't have sound and so on. And they found that a part of the brain called the insula, which you can see here, lit up. That is, when they compared different states and subtracted them, there was a difference. And because a few studies have shown that maybe the insula has something to do with being in love, they therefore concluded that people are in love with their iPhones. We understand love. It extends even to devices. But of course, there are many, many, many problems with this study. And the publishing of this article launched many responses from angry scientists who disputed the conclusions. And one obvious example is that the insula is implicated in many different behaviors, not only in being in love, but also in the response of disgust, and also in hunger. And so it's really going a bit too far. Okay, here's another one from Discover magazine. Eating cheese and meat may boost self-control. So, okay, we clearly want to understand self-control. You know, there's something that it means to be in control and something that it means to be out of control. And if we understood how the brain did that, that would be intellectually interesting. It would help us a lot. Sometimes people lose control when they don't want to. And so some scientists studied this, and they had some people in volunteers in the lab, I should say, and they gave them a substance that messed with their neuromodulators a little bit and found a small difference on one task involving revenge. And because the But it was like the lab version of revenge, where like you had a partner who didn't get a quarter because you were mad at him or something. You know, it was a very contrived situation, and the researchers were very upfront about the nature of their study. But nevertheless, it was taken to extreme, and because the substance that was in the drink was tryptophan, which you also find in cheese and also in turkey, then the press just went wild with this and suggested that, you know, if you have any self-control problems, a grilled cheese is really where it's at. Or even, and this was actually the image from the Discover magazine, a turkey. So if any of you are feeling out of control, I recommend you run down to the deli and get yourself a turkey sandwich in short order. It might help things. So, okay, so now why is this such a hard problem? You might wonder, what is taking neuroscience so long to get to the bottom of these issues? After all, this is just, a part of the body. There are other parts of the body we understand very well. We understand the kidney well, we understand the liver well, and so on. What's going on with the brain exactly? And I'm going to argue that there are really three main reasons why this has been a very difficult problem to solve. And the first reason is that brains are very complicated. So what I've shown you here is an image of a mouse brain, which is much less complicated than a human brain. You'll perhaps be reassured to know. And you can see many tiny little dots, each of which is a neuron. These are stained with a particular stain that makes neurons turn purple. So if we zoom in a little bit, you can see that there are many, many, many neurons, and there's a complicated laminar structure. And now when I really zoom in, you can start to see, even in this tiny, tiny section, which is so thin, it's about the thickness of your hair. Very, very, very thin. You can see there are so many neurons in here, and that makes things really very difficult to understand. So in the human brain, we have many more neurons. We have about 86 billion neurons in our brain. And so to try and understand how they all work together is a very big problem. And to make matters worse, different parts of the brain have very different structures. Here I've put two parts of the brain next to each other. On the left, you see a slice of the cortex, and that was taken from a mouse brain that was sectioned like this. We call this a coronal section. And if you keep going further and further back, you get to what's called the cerebellum, and that's what I've shown here. And what I hope is evident is that these two parts of the brain, which are from the same animal, have very different structure. And this makes the problem only all the harder. Not only are brains complex, but they differ as you go from one part of the brain to the next. So that's the first problem. The second problem is that introspections about brain function are misleading. So what do I mean by that? So when you think about the brain, it's tempting to try and understand it by just thinking about it in your mind, right? You're a person, you have a brain, you think, well, maybe if I ponder it in my mind, I'll come up with some good solutions. And the problem is that this can be very misleading. And you've already had an example of that earlier in the talk when I mentioned Empedocles, the Greek, who had an idea about how vision worked. And his idea based on introspection was that light came out of the eyes and bounced off of objects. And indeed, if you'd never done an experiment, this was potentially a reasonable explanation of vision. But if you go into the lab and do experiments, then it immediately becomes obvious that your introspection was incorrect, and that in fact it works the other way around. The light comes into the eye and hits the photoreceptors that are on the back of the retina. So that's the first example. A second example of how misconceptions about brain function can be misleading has to do with, as a field, which problems we thought were going to be easy to solve and which ones we thought were going to be difficult to solve. So for example, if you think about really kind of the peak of cognition, what's something that only humans can do, and not only that, but really, really smart humans. Well, what's an example of that? Well, when people thought about this at first, they thought, well, what about chess? It's a hard game. Mice can't play chess. Little children can't play chess. It takes a lifetime to master. This has got to be the workings of some seriously advanced machinery that we may never understand. But at this point, we have computers that can beat the very best chess players in the world. So even though it seemed like the algorithms required to play chess were so incredibly complex that we'd never be able to solve them, this turned out to be a tractable problem that we worked out about 10 or 15 years ago. Now, by contrast, the ability to actually, so we'll put the computer ahead on that front. The ability to actually move the chess pieces around, now that's a completely different story. So we have no idea how the brain is able to pick up a chess piece and hold the fingers in just the right spot and move it to the right place. And to build robots that can do that is extremely difficult. We're nowhere close in our robot technology to even the level of a small child. So it seemed like that would be an easy problem. We move our bodies and move objects around effortlessly every day. But to understand how the brain does that has been incredibly difficult for neuroscientists. And just to make that point again, I'm going to show you a movie of the state-of-the-art technology in robotics for manipulating objects. In this case, you're going to see a robot manipulating a cup. So you'll see the robot's hand come down. And this is hard, right? Because the robot has to find the edge of the cup, move the fingers, and just, all right, wow, it's hard, right? It's hard. Come on. Yeah, so finally he got his hand in the right spot. Pretty good for the robot, not bad. Okay, let's see where things are. Oh, there he goes. He even lifted it up. Let's see how things are. Oh, come on, keep going, keep going. Okay. Thank you, robot. Okay, let's see how things are, not in terms of robots moving cups around, but humans moving cups around. What's the state of the art in terms of humans moving cups around? Well, let's see. She won the cup stacking world record. So you can see that the humans are way, way, way ahead of the robots when it comes to stacking cups. And so this makes it clear that our intuitions, that is our introspection about what's a hard and difficult problem to solve in terms of neuroscience, just turn out to be wrong. We got that one wrong, and that happens all the time. So that was the second reason it's a hard problem. And the third reason why it's a hard problem is that brains do different things in different animals. So consider for a moment a mouse and a bat. So these are both mammals, and in fact, they're both rodents. They're actually pretty closely related from an evolutionary point of view. But think about what the brains of these two animals have to do they do very different things. So if you're a bat, you have to have a motor cortex that can make you fly, to move your wings in just the right way to fly above the ground, and a mouse obviously has none of that. And also, bats can navigate via echolocation, so they need to have very special neural machinery that allows them to process the incoming sound signal so that they can figure out where things are in the world. And mice have nothing like that. And these are just two examples in two very similar animals. If you think about the whole range of animals that there are in the world, the problem escalates even further. And the reason why this is such a challenge is as follows. In general, the approach that biologists have taken to understanding physiological problems is that we look to see how these problems are solved in animals. So when people wanted to understand, going back again to the liver and kidney, the internal organs, they did so by looking at the livers and kidneys of different animals, of cows and pigs and mice and so on. And even though the kidneys of those animals aren't identical to human kidneys, they're pretty much close enough. And that's why medicine has been able to make so many advances in terms of other kinds of organs through the use of animal research and using animals to understand those organs. But when it comes to the brain, as neuroscientists, we have to be really, really careful, because any animal that we study will necessarily have a brain that's different from ours, because each animal is adapted for a particular biological niche. And so as scientists, we have to pick our model system, that's what we call an animal, that we use to understand a process, very, very carefully, and acknowledge that there are always going to be key differences between that animal that we study and ourselves. So that's the third reason why it's a challenging problem. Okay, so I've told you so far that there's a gap between our understanding of sensory motor reflexes and truly complex behavior. And I felt like maybe it was bad to put the complex behavior on the Manhattan side, so I'm going to switch that. And I've told you that this gap exists for a few reasons. Brains are complicated, introspections are misleading, and the brain does different things in each animal. And when I started my lab, my goal was to bridge that gap. I wanted to bridge the gap between complex behavior and sensory motor reflexes. And I thought a little bit about how to do that. And I thought about, well, what is it that makes a behavior more complex than something simple? And I thought about a few key features. So first of all, think about the knee-jerk reflex that you do when you go to the doctor. There's no flexibility in terms of the timing of that reflex. The doctor hits you with the hammer, your leg goes up, it happens exactly when it happens. But with more complex decisions, they operate on a much more flexible time scale. So for example, consider the decision to buy a car, right? You go out, you learn about the cars. You can make that decision at any moment. It isn't like the sensory motor reflex where you're locked into a particular timing of a sensory input that requires you to make a decision. You can make the decision to buy a car at any moment. You might see one car, you might be indecisive and see 100 cars, pissing off your partner to no end. But it operates on a flexible timescale. So that was the first difference. And the second difference that I thought was a key one is that complex decisions integrate many different sources of sensory information. So again, think of the car. When you decide to buy a car, you use your visual system to look at the car, the olfactory system to smell the car. You might then take the car for a test drive and use your auditory system to listen to the car's engine to get a good sense of whether it's a good car or not. And you put all of those sensory inputs together, you integrate them to make a decision. And so for me, this seemed like really a hallmark of more complex behavior that might allow me to start to bridge the gap between simple sensory motor reflexes and complex behavior. So when I was starting my lab, I wasn't quite sure how to go about doing this, and I wondered whether there was a lot of work already on understanding the neural mechanisms of this process, which we call multisensory integration, because you're integrating sensory inputs from multiple modalities. And it turned out that there really were only a few groups who had studied how the brain puts together multisensory inputs, which I thought was sort of surprising. But interestingly, there was an extensive study of the behavior, and the behavior turns out to be really, really interesting. So, for example, this is a picture of a ventriloquist, and you've probably all seen a ventriloquist. This is a multisensory illusion because your brain gets tricked. So let me just explain. So in a ventriloquist, the sound is coming from the speaker's mouth, but it appears to be coming from the puppet's mouth, even though the puppet's mouth is about 30 centimeters away from the location of the sound. And so as an observer in the audience seeing a ventriloquist, you're not at all bothered by the fact that the sound is coming from a different location from the puppet's mouth, because the movement of the puppet is so compelling that it fools you into thinking that that's where the sound is coming from. And the reason for this has to do with the nature of the auditory and the visual system. So our auditory systems are not very good at spatial localization. They aren't really designed for that. So if you only have your ears to go by, it's hard to tell where things are in space. But your visual system, especially because all of you are primates, your visual system is excellent in terms of spatial resolution. Using your visual system, you can pinpoint something exactly in space. And so in the case of the ventriloquist, even though the sound is coming from the wrong place, you aren't bothered by it because your visual system is so reliable in terms of spatial precision that you just go with the visual system and you just aren't bothered by the auditory mismatch at all. And in fact, all of you right now are experiencing what we might call extreme ventriloquism, because right now I'm talking here, my lips are moving here, but the sound is coming from the speakers that are over there. But I doubt very much at the beginning of the talk that anyone was kind of bothered by this. It was like, whoa, she's talking here, but the speakers are there. Like, oh my God, it's crazy. It's a multisensory mismatch. No. Because your auditory system doesn't really know where the sound is coming from anyway. And the fact that it's coming from 10 feet away from where I'm talking doesn't bother anybody. We're used to that and it doesn't bother us. And you might be tempted to think that this is because we're very visual creatures. We're primates, we have great visual systems. And actually that's what a lot of people did think for a long time. But it turns out that there are other multisensory illusions in which the reverse happens. Okay, so here's an example of one of those. Okay, so now we have some audience participation here. So your job as the audience is to say whether you see one little black circle or two. So one flash or two. Okay, you guys ready? One or two? Two, yeah, everybody always says two. Okay, how about over here? Oh, yeah, somebody said one. Okay, how about over here? One. Okay, so now I'm gonna play those exact same movies, but this time with the sound off. Okay, so start by looking over there. Oh, so it was only one. Okay, and this one was only one. So what happens is because there are two beeps, your auditory system tricks your visual system into thinking that there are also two flashes. And what's interesting to me about this illusion is that it's so compelling that even once you know about the illusion, you really can't suppress it. Okay, so I'm gonna play it one more time. It really looks like two. It really looks like two, but it's actually one. And this is because when it comes to timing information, your visual system is really not very good at all. Our visual systems are slow. The machinery that turn light into electrical signals are very sluggish. And so the visual system, even though it's good at telling you where things happened, is very bad at telling you when things happened. And the auditory system, even though it's bad at telling you where things happen, you don't even know where the sound is coming from, it's very good at telling you about when things happen. So because this particular illusion depends on very precise timing, then your visual system gets fooled by the auditory system. So from these two examples, it might be tempting to infer that multisensory integration is kind of an all or none thing. You either go with the visual or you go with the auditory. And indeed, that's what people in the field thought for many years. But it turns out to be kind of more subtle and really much more interesting than that. And that was made clear by some experiments that came with sort of a strange drawing, but were really very interesting experiments studying how subjects integrated multisensory information that was visual and also what's called haptic. And that means when you feel something with your fingers, that's haptic information. And so what they did is they had subjects judging the height of a raised bar. And they would either feel the raised bar with their hands, as the subject's doing here, or they would see the raised bar that was computer generated from what's called a random dot stereogram. So the scientists showed two random dot patterns, one to one eye and one to the other eye. It's kind of like when you go to a 3D movie, and the subjects would have the illusion of seeing a little 3D bar. So they'd see it with their eyes, and they'd feel it with the hands, and they'd have to make a judgment about the height of the raised bar. So not surprisingly, the subjects did better. That is, they made more accurate judgments when they had the multisensory information. That is, when the visual and the haptic were presented together. But what was surprising and really kind of put these guys on the map is they found that this integration is done in a really smart way. That is, it depends on the reliability of each stimulus. So the way they figured this out is they took those random stereograms that they presented to each eye, and they sometimes messed them up. They added a bunch of noise so that all of a sudden the visual stimulus wasn't very reliable anymore. And they found that under those circumstances, the subject would tend to rely more on the haptic information. But when they then changed things so that the visual was again reliable and subjects had a nice clear picture, they went with the visual again. So it seemed like the brain was able to, even on a moment-by-moment basis, change the way that it integrated the multisensory information. And in the details that I won't go into too much, they found that subjects, these are just human subjects, undergraduates from Berkeley, they found that they approximated statistical optimality. That is, they weighted each stimulus exactly as much as they should, given how reliable that stimulus was. So to me, this was a really exciting finding, and it seemed to me the sort of thing that would make it possible to bridge that gap between sensory motor reflexes and complex behavior. Because here's a behavior that's putting together multiple sensory inputs, and it's doing this in a very smart way that's taking into account the statistics of the environment. So I decided this is it. This is the problem I want to work on. I want to go in and tackle this and find the underlying neural mechanisms that make it possible for the Berkeley undergraduates to optimally combine multisensory information. Now, the challenge was I wanted to do this in a model animal where I could record neurons, and also one where there were powerful tools available of the sort I alluded to at the beginning of the talk. So when I started my lab at Cold Spring Harbor almost exactly 3 years ago, I decided that I wanted to do this statistically optimal multisensory integration behavior in rodents. And I should say that at the time, there were a lot of skeptics. People said, okay, no, I know that rodents can navigate, they can do certain things really well, but come on, statistically optimal multisensory integration in a rodent? I mean, that's crazy. There's no way that the rodents are going to do this. It's not a good idea. But I was undaunted and went out to start my own lab nevertheless. It was a pretty risky thing to do because I really didn't have any idea whether these animals were capable of this kind of behavior or not. But I reasoned that putting together multisensory information is something that animals have to do in the wild all the time. And so probably all animals evolved to have the machinery that makes this possible. So I was fortunate in assembling some other like-minded scientists who were likewise willing to come and work with me in my lab and take this risk to try and figure out whether we could drive, or rather elicit, this really interesting behavior in rodents, and then allowing us to take the next step in the journey to go after the neural mechanism. So the first thing we had to do was figure out a way to test for this kind of behavior in the rodents. Obviously, it wasn't going to work to have this sort of a task here. We needed to design a new one that was more suitable for what rodents are really like. So this is what we did. So in our task, the rodents in our lab, their job is to make a decision about the rate of a stimulus. That is it flashing and beeping at a high rate or a low rate? So when they come into the lab each day, they approach this little device, which we call a behavior box, and it consists of 3 ports. These are little nose pokes. They can put their nose into there, and then there's the port will detect their presence. And then there's a panel of flashing LEDs to present the visual stimulus, and then also a speaker. So the rat will approach the center port, and then after he does that, this triggers the onset of a visual, or sometimes auditory, or sometimes both together stimulus. And the job of the rat is to report whether the stimulus is at a high or a low rate. Now, of course, the rat coming into the lab, or the mouse coming in for the first time, has no idea what high versus low rate means. This is something that we as experimenters define. We pick an arbitrary number as our cutoff, and we say, okay, below this is a low rate, above this is a high rate, and the animals simply learn that with experience. Okay, so for example, I'll now show you a low rate trial. So for that kind of a trial, the animal would be rewarded for going to the left, and he would get a drop of water for making a correct choice. And now I'm going to show you a high rate trial. And for that kind of a stimulus, the animal would be rewarded by going to the right. So how do we quantify this kind of data? Before I show you that, here's a movie of an actual animal in the lab who's doing the task. So this is the center port here that you saw in the schematic a moment ago. And these are this is where the visual and auditory stimulus will appear. And here are the 2 choice ports where the rat will make his decision. So sometimes you see he gets a visual by itself, and he's not bothered by the fact that some of them are visual, some auditory, some multisensory. And these are pretty easy trials, but he gets every single one of these correct. They're all correct. He gets water on every trial. Okay, so you might wonder how we quantify this behavior. The way that we do that is to consider the rate, that is the number of stimulus events that are presented over the course of a 1-second trial, and the proportion of the time that the animal makes a high rate choice, that is the proportion of time that he goes to the right. Okay, so for example, suppose we present a low rate stimulus. The proportion of high rate choices is naturally pretty low because that was a low rate stimulus and the animals are well trained. So he would pick the high rate choice only very rarely for such a stimulus. Now here's a high rate stimulus. And a well trained animal almost always makes the high rate choice for stimuli like that. Now here's the interesting part. We take stimuli that are intermediate between those two extremes. It sounds a little like Morse code. I'll play that for you again. So this stimuli has a rate that's intermediate between the very highest and the very lowest rate, and the animal picks the high rate choice about half of the time. So half of the time he interprets that as low, and half the time as high. And this particular value here, 12 events per second, that's the arbitrary category boundary that I described a moment ago. This is a value that we set, and what it means that when the rates are below that number, he's rewarded for making low rate choices, and when the rates are above that number, he's rewarded for making high rate choices. So we can collect lots of data from a particular condition. This is data in response to auditory only. That means these are the animal's decisions when he heard the clicks but didn't have any visual stimulus at all. And here's the same thing for the visual only, so only the flashing LEDs and no clicks at all. And our question was, can the animals make more accurate decisions when they're presented with multisensory information as opposed to just visual or just auditory alone? And what that would look like is it would mean that this curve for multisensory would be steeper than either the auditory or visual alone, because a steeper curve means more correct choices. And for this animal, this is the data for the multisensory version of the task. So you can see that their accuracy is improved. Yay, rats. So we were very happy to see this improvement in the rats. And I should say that you might notice that our task is really a very unnatural task. It's nothing like the rodents would encounter in their normal environment. But nevertheless, after we train them on the auditory and visual version of the task, we typically find that as soon as we introduce the multisensory stimulus, that we usually see this improvement right away. This isn't something we have to train in the animals. And this suggests to us that even though the stimulus we use is very unnatural, that it's tapping into neural machinery that is already there and exists presumably to help the animal integrate multisensory information in a much more natural context. So I should say that this data was collected by my student. Unfortunately, the color is bad on the projector. My student, David Raposo, who's not just a shadow. He's an enigma. But he's here today in the audience, so at the end of the talk, you can ask him questions. Okay, so now you've seen what the rats do in the lab, and so now you can have a chance to be a bit of a rat yourself and see what the sensory experience is really like. So this would be an example of a high rate trial. I'm going to show you a high rate and a low rate, and then you guys have to guess about some intermediate ones and see if you can figure it out. Okay, so this is a high rate stimulus. Okay, did everybody get that? That was high? Okay, now here's a low rate stimulus. So those were the two extremes. Now I'm going to do one that's in between, and you guys are going to have to report whether you think it's high or low. How many people think high? How many people think low? Yeah, I can't tell. I don't know. It's intermediate between the two. It's very hard to tell, and you guys were 50-50 even split. And there was one last stimulus that was, again, very close to the category boundary, but had even much lower contrast. So this gives you a little bit of a sense of what it's like for the rats. But I'd like to collect some actual data. And so I'd like to invite someone from the audience to come up and just for a moment to be a rat in our lab and to collect some data on the task. So do I have any volunteers? Any of you? Don't be shy. Don't be shy. Okay, you, come on up. Come on up. I don't know how you should. Oh, you have to follow the way around. Yeah, you can follow the way up. Okay, welcome. What's your name? Isaac. Okay, everybody give Isaac a hand. Okay, come and have a seat. You're going to put these headphones on. Let me just enter your name. Okay. All right, so have a seat. So it's going to give you instructions. So once you have the headphones on, you can just follow. Okay, very good. See how Isaac does. Okay. All right, so he's going to do some trials. They're giving him instructions there. Is it? Oh, yeah. This one is blue. Yeah, it's hard. The light is weird. Yeah, that one's yellow. Okay. Yeah, you're good? This guy is pretty brave. I'm impressed. Okay, so while he's doing that, I thought you guys might want to know a little bit about how we train our animals. You might wonder how we take these... Go ahead. Oh. Yeah. Okay, so you might wonder how we get them from being, rats off the shelf to being exquisitely trained to make decisions about stimuli that are very close to threshold. And so my postdoc, Matt Kaufman, who's also here today, sitting right there, made a video to try and make it clear the training process that the animals experience. Okay, so Isaac has completed his task here. So how did you do? What was your percent correct, Isaac? High B. Okay, so I see that he got an 88% correct. It's pretty good. It's pretty good. Let's give Isaac a big round of applause. Okay, hang on. Don't go yet. Don't go yet. Wait. I have, just while you wait, here's a little cheese. Okay, so we're just going to run a little Python script here. Okay, good. So now we can see how wow, this is amazing. We can see how Isaac did. He did better than our best rat. Isaac, that's Isaac. Wow. Amazing. This is really a select crowd you guys have here. I'm very impressed. Okay, so he did very well. Typically the humans do slightly worse than the rats, except for Isaac. Okay, all right, so I've told you now a little bit about the behavior that we study in the lab, and you've got to experience it at least a little bit, or at least Isaac has firsthand. So, in our lab, we're really pretty excited about the behavior. We think about it a lot, and we think you can learn a lot about neural mechanism just by studying the behavior. But really, for us, the behavior is only the first step in the journey. And having developed a behavior that invites animals to integrate across time and integrate across sensory modalities, we're now in a really strong position to go into the brain and to really try and understand the neural mechanism that makes this integration possible. And when we set out to do this, this step is the part where we can begin to take advantage of the emerging technologies that I've been mentioning throughout the talk that make this such an exciting time in neuroscience. So when we were designing this experiment and trying to figure out what's the right thing to do to understand the neural mechanism, we took advantage of these emerging technologies really right from the beginning. So the first thing we needed to do was to figure out where in the brain to record. That is just to study, to try and find the neurons that play a role in this behavior. And one fact about parts of the brain that's informative about the roles they play in behavior are the other parts of the brain that they're connected to. So a first step that people will often take is as follows. So they will take a virus and inject it into the brain, and viruses infect neurons and can even travel from one neuron to another. And if you hook up a fluorescent tag to the virus, you can then look at the sliced tissue under a fluorescent microscope and see which parts of the brain are connected to which other parts of the brain. And this can be really helpful in identifying candidate areas of the brain to look for neural mechanisms that are associated with particular behaviors. So for example, a candidate area for us was the posterior parietal cortex, or for short, the PPC. And it turned out that people had made such injections into PPC. And in the traditional technique, at the end of the day, you get an image that looks something like this. So each place you see a dot is the place where, after an injection into PPC, remember that's the posterior parietal cortex, that they saw some expression in a fluorescent microscope. So this dot means that this part of the brain is connected to the PPC. And this was kind of a good start, but the problem was that, remember that these sections like this is one slice taken from a brain, and this slice is flat. But the brain, of course, has three-dimensional structure. And in the olden days, what people would do is they would look at many such slices like these, taken from different parts of the brain, and try to kind of mentally reconstruct these dots into these 3D pathways. But the truth is that it never really worked very well, and you were never left having a real sense of what the 3D structure was actually like. But now, fortunately, thanks to some software development, there's a new tool that makes it possible to do that very easily. So things start off the same way. I've positioned this syringe above the PPC, and just as before, people can inject different substances in, and every time you see a dot, that means that the PPC projected there. But here's the cool part. Many such images can be put together, and then the brain can be rotated arbitrarily in any direction that you want to visualize the full extent of where those projections go. And this changes everything, because it means for the first time, we can see the brains for how they really are. That is, three-dimensional structures with connections that are all over the place. And when we use this software, which I should say was developed by the Allen Institute for Brain Sciences, and this is a really interesting organization. They've made all of their brains and also all of the tools to manipulate the brains, like the movie that I made to show to you tonight, are freely available online. And it's a really powerful resource that has really helped a lot of us in the field. So that's kind of the first example of an emerging technology that's making neuroscience at a really exciting moment. So through this kind of visualization, we realized that the PPC was a good place to look because it was heavily interconnected with visual and auditory areas and also projected to the frontal cortex and parts of motor cortex. So here we are back to the flattened 3D version again. And remember, this black square is over the PPC for posterior parietal cortex. And of course, there are millions and millions of neurons there. But imagine just a few. Here's just a few neurons here. And the neurons in the brain communicate with each other via electrical signals called action potentials. And so different neurons in the brain are sensitive to different sensory stimuli. And when you present sensory stimuli to the brain, the neurons fire action potentials in sometimes complicated patterns that are difficult to understand. And what we can do is to put an electrode in the brain, just a piece of metal wire, and record the electrical signals, that is, these action potentials, from the neurons to try and understand what they're doing. And this approach, which relies on standard off-the-shelf technology, we're just amplifying small electrical signals, can give us insight into what the brain is doing. So in this case, our hypothesis is that PPC neurons reflect developing decisions. So for example, a signature of this hypothesis might be that PPC neurons fired only a few action potentials into, say, those low-rate visual stimuli that you saw, but lots and lots of action potentials into the high-rate stimuli that you saw. So, as I mentioned a moment ago, this is conceptually pretty straightforward. It's an electrical signal, you amplify it in a conventional way. But the challenge is that we want to record these signals from one neuron at a time so we don't get the signals mixed up. And we want to do this in the context of an animal who is awake and moving around and completely unrestrained, as the animals are in the movies that I showed you a moment ago. And this is the really challenging part. So in my lab, we build what we call tetrodes. We take tiny, tiny, tiny little wires, 15-micron wires, thinner than your hair, and we braid them together, and then we put them in a little device called a microdrive, which we can advance into the brain. And it's only this tiny, tiny little bundle of 15-micron wires. That's the only part that goes into the brain. And so this doesn't cause any stress or harm to the animal because you can't feel when something is inside your brain. There are no sensory nerves there that can detect things. And it does almost no damage because the wires are so tiny. So we then take a bunch of these microdrives and put them all together in a little holder so that we then, at the end of the day, have 8 microdrives, each with 4 wires, 4 electrodes in them, and we can record activity from lots and lots of neurons as the animals are engaged in behavior. And so for us, as electrophysiologists, that is, people who record electrical signals from cells, what we do in the lab, what we spend a lot of our day doing, is we get the animal hooked up and we sit there and we watch the animal and we listen to the cells. And it's by listening to the cells that we can gain insight into what the brain is actually doing and to try and understand whether it is indeed a candidate area for a particular process that we're interested in. So just so you know what it's like to be an electrophysiologist in our lab, we might sit watching the animal do behavior, which I'll show you, and listen to some spikes, which I'll play for you. So this is a very short clip, but every time you hear a sort of a bursting clicking sound, that's the response from a single isolated action potential that was recorded by my student David in the lab. So we can listen to the animal's neural activity as he's engaged in the task. So we do that many, many times. The animal does many of those repetitions or trials, and we average them all together to generate responses that look something like this. For this particular plot, I've grouped the trials by what happened and color-coded them, so that in this gray trace here, this is the response of a single neuron to a low-rate stimulus, and this is the response of a single neuron to a high-rate stimulus. And you can see that this neuron distinguishes the 2 kinds of stimuli, starting pretty early on. So the stimulus starts here when I put time equals 0, and about 200 milliseconds later, the neuron does something very different in high-rate trials versus low-rate trials, in keeping with our idea that the PPC plays a role in developing decisions. And here's a neuron that likewise distinguishes the 2 stimuli, but has the opposite profile. It responds more for high-rate stimuli than for low-rate stimuli. So these experiments are a really good start because they allow us to correlate the neural activity that we measure with the animal's behavior. But at the end of the day, it isn't really quite enough because we need to go a step further and perturb the neural activity and demonstrate an effect on behavior. And through this kind of experiment, we can show that this area plays a causal role in driving decision making. And fortunately, where we are in neuroscience, we're in a really good position to do that with more precision than anyone ever has before. And that's thanks to the development of some new technology. So remember, we can record from neurons in the brain. And there's a new technology that was developed about 10 or 15 years ago called optogenetics. And the idea is that you take a virus, similar idea to the virus before, but now it delivers a very special light-activated ion channel called channelrhodopsin to all the neurons. And this channel will be incorporated into the neurons. And when experimenters later do something which seems very arbitrary, they shine blue light on the neurons, all of a sudden the action potentials will go from their normal low rate to a really, really high rate. So using this technique, we can very precisely perturb the activity of the neurons and then see whether this has any effect on behavior at all. So our experimental design was like this. Our hypothesis, again, this is the same hypothesis as before, is that the PPC plays a role in decisions. And here's our prediction. We think that because we only had the blue light on one side, that shining the blue light during the decision will cause the animal to make more choices in one direction than in the other direction. That is, he'll develop a bias for decisions in a particular direction. And if we're wrong, if PPC either does nothing for decisions or is just along for the ride, then it won't matter whether we shine the blue light or not. The animal will always make the same decisions. So here's some control data for not the laser yet. Laser's still turned off. This is just the decisions the animal makes in response to the visual system, the visual stimulus. And I've plotted again the proportion of high rate choices. And this animal looks like he prefers low rate a little bit because he only picks high rate about 30% of the time. And so if shining this laser on the neurons causes the animal to make more high-rate choices, then the number of high-rate choices during laser stimulation should be much higher than for the control trials. And I'll show you that data now. So you can see that the animal makes many more high-rate decisions when the visual stimulus is paired with the optical stimulation, the laser, than when the animal is presented with the visual stimulus alone. And this is really compelling evidence in support of the hypothesis and is in keeping with our prediction. And this work was done by my student, John Shepard. So I started off telling you at the beginning of this talk that there's a big gap between our understanding of complex behavior and our understanding of sensory motor reflexes. And I've now taken you on a journey through the short history of my lab and told you about how we think that decision making can begin to bridge that gap. So, have we bridged that gap yet? Well, I think we've made an important first step by studying decisions that integrate all kinds of different sensory modalities. But the truth is that my lab and also the field of neuroscience, we really still have a long way to go. For example, what happens if you're integrating not just two sensory modalities, but maybe a sensory modality and a memory, or a sensory modality and an innate drive? Or what about decisions that integrate information not just over the course of a second, as we do in our laboratory, but over the course of a week or a month or even a year, as some real-world decisions actually do? So many aspects of those complex behaviors still are mysterious, but our hope is that by understanding the simpler decisions that we understand in the lab, we'll uncover mechanisms that are relevant to complex decisions as well. And the mechanisms might not be identical, but the computational principles may be conserved as we go from sensory motor reflexes to more complex decisions all the way up to complex behavior. So before I take questions, I should acknowledge the people in my lab who did this work. I've mentioned some of them today. David Raposo and Matt Kaufman are here in the audience. We also have a lab blog, if any of you would like to check it out. We write about things that we're doing in the lab and also things that we're thinking about. And I'd also like to acknowledge my funding sources. As many of you probably know, neuroscience and basic science in general is at a difficult time in terms of securing enough funding to do the work that we do. So my lab is really very grateful to the financial support that we have from these organizations and also from Cold Spring Harbor Laboratory. So thank you very much for your attention, and I'd be happy to take questions.